Answering the age-old question, if you pop a human brain organoid into the skull of a mouse, does it change its behavior? Yes, and this paper is so wild. What they did in this adorable schematic is took cells from people with Down syndrome and people without, and then induced them to become the little brain organoids and popped them in the skulls of mice, right onto their brains. Our little brain friends are, by the way, almost always made from donated skin tissue and not fetal derived. These ones are made from blood cells. But back to what happens if you want more brain per brain. Brain squared? If you're wondering how that works, you also have to know that the brain organoids are quite invasive. We've discussed this before, but they'll make hybridized connections between even different organisms, between rats and mice or human. They're essentially at a very, very early stage in development. That means that they can grow and develop connections that you might not expect in an adult brain. And no, they don't just take on the same structures as their host, they're still going to look like the original organism that they came from. And that is our little mousy forebrain after 6 months. What they found in these mice was overproduction of OGL2, which is involved in the proliferation of motor neurons, so overproduction of it could cause some problems. This gave them a target for how some of the cognitive issues that we see in people with Down syndrome could be treated, but it goes much farther. Finding the upregulation or downregulation of genes involved with cognition and memory could help with other diseases and even inform us of potential targets we're not even aware of.